In our cells, proteins physically interact with each other as part of the functions they perform. As an example, proteins often join together to form large protein complexes that perform important biochemical reactions, like producing cellular energy in the form of ATP molecules. To visualize this, here are three proteins called F, G, and H. Proteins F and H fit together, for example, to produce ATP molecules. Protein G is not involved in this process, so it can be ignored for now. What might happen if changes, mutations, occur to the DNA sequences, genes, that encode these proteins? Those mutations can cause the shapes of the proteins to change in such a way that the new mutant versions cannot physically interact in the same way. Sometimes these protein structure changes render them unable to perform their usual cellular functions. In this example, when a mixture of the original gray proteins are present along with the new red and blue versions, the essential protein interactions can occur. However, if an organism only had the red and the blue versions of the proteins, then its cells might be very unhealthy. So, it is important to understand how genetic changes can cause changes to proteins and how those changes can influence the cell and the organism.